Good morning. How are you? Fine. Okay. It's a good day. Uh, so today we'll start topic. So let me know about our previous lecture. Anyone in the class? What was our previous topic? About antibiotic. Okay, dear student. So who will define antibiotic? Anyone in the class? Please stand up. Okay, good. Which stop or kill the microorganism, especially if we can say it may be bacteria. What was the first antibiotic among female? Penicillin. Okay. So the first antibiotic was a penicillin, and it was obtained from fungi. Do you know the name of the fungi from which this antibiotic was obtained? Do you know? It was Penicillium notatum. Penicillium notatum. And who discovered this antibiotic? It was Fleming in the year 1928. So this was little about our previous lecture about antibiotic. You are the student of class second year pre-medical. So we will slightly move into the depth of antibiotic. Further, we can classify antibiotic based on its inhibition, that how it kill or inhibit bacterial growth. So antibiotics, they destroy or inhibit the growth of other pathogenic organism, microorganism. It may be fungi, it may be bacteria. But today we will slightly move and discuss something about bacterial inhibition or killing. We have classify bacterial, the antibiotic used against bacteria and how it works. What is the mechanism of action? The mechanism of action. So, dear students, you know that bacteria is actually what? It is aprokaryotic, while human being is a eukaryotic. So we will target the organelles, the site, the parts of microorganism which is not common. So you know that cell wall is present in bacterial cell, but it is not present in human cell. So the first choice is the cell wall synthesis inhibitor. How does it work? There are many antibiotics. For example, I will give you one example of Silval inhibitor. For example, cephalosporin. Cephalosporin. This antibiotic, how does it work? It works by disrupting the bacterial cell wall. How? Because it inhibits the enzyme which is used in the formation of cell wall. You know that bacteria cell wall is made of what? Do you know? Peptidoglycan. Peptidoglycan. Cell wall is made of the substance present in the bacterial cell wall is a peptidoglycan. And you also know that in gram-positive bacteria, this peptidoglycan layer is a thick, while in a gram-negative bacteria, it is a thin. So this cephalosporin inhibit the enzyme. When this enzyme is inhibited, the bacterial cell wall is inhibited. When cell wall do not form, it eventually lead to the lysis of a cell wall and eventually the cell death. So this is the first antibiotic used against bacteria. And the next option, we can also target protein synthesis of bacteria. For example, tetracycline. Tetracycline, it is an antibiotic. But you also know that there is the ribosome of bacteria is formed of two units, 30S and 50S. 50 said work. These are units which collectively form how much? Do you know? 70. 70 said work. But actually what? Tetracycline only target 30 said work. It is one thing. One thing is more important part. You should know that 30 said work unit is not present in human cell. 30 said work is the part of ribosome, but this part is not present in a human cell. The binding amino acid tRNA, and thus actually what is called protein synthesis. Protein synthesis, a terminology used for it is called translation. When DNA is formed from DNA, it is called replication. When RNA is formed from DNA, it is transcription. And when protein is formed from RNA, it is called translation. So actually, translation process is stop which inhibit the growth of bacteria. The third option which we may target bacteria is DNA synthesis inhibitor. Metronidazole is an antibiotic stop protein synthesis by interacting with DNA synthesis. How? And cause losses of helical structure. You know that actually DNA is a double standard. DNA is a double standard. DNA is and it is helical. A stable structure is helical. Helical structure. So this antibiotic laws of helical structure, it disrupts the helical structure. Once this helical structure is disrupted, so it causes the cell death. So it was the third antibiotic, cell wall inhibitor. We may target bacteria, bacterial protein synthesis in the shape of tetracycline, which target 30 Swedberg. We have also option. We may target the 50 unit also. And the name of that antibiotic is called, which is available in the market, macrolides. Macrolides. So we have plenty of option to treat the infection and bacterial, the pathogenic bacteria. We have plenty of option. But one thing is more important. I will teach you in the last this that self-treatment is strictly prohibited. The last but not the least, RNA synthesis inhibitor. We know that bacteria is what? Bacteria is actually a cell. So we may also target its RNA. For example, RNA, RNA polymerase. RNA polymerase, it is the enzyme which actually what it do? It causes polymerization of RNA. You know that, that RNA is form of nucleotide. So when such an antibiotic is used, we can target its protein, we can target its bacterial DNA, and we can target its RNA synthesis inhibitor. Bacteria can be inhibited by what? By its RNA. For example, ribomycin, which stop translation. What is translation? Translation means protein synthesis. Protein synthesis. So when the protein synthesis of bacteria is inhibited, this antibiotic is called bactericidal. Some antibiotic is bacteriostatic, while this antibiotic is a bactericidal. Last is my opinion about antibiotic. You should learn in detail. You can also visit and search in detail about my recent publication article, which is available. Antibiotic resistance and hemodialysis patients. It is my online article publication, which is online available. Antibiotic resistance and hemodialysis patients with my name, Zaid Basha. So dear students, it is an international journal. You can find with this name, antibiotic resistance and hemodialysis patients, with my name, Zaid Basha. You can search in detail, you can study in detail such a topic, antibiotic. And let me add something from my side of that it is estimated, especially in developing countries like Pakistan, that there is plenty of drug resistance. So, so in developing countries like Pakistan, there is extreme drug resistance. There is a extreme drug resistance. So before taking antibiotic against any infection, we should avoid self-treatment. We should avoid self-treatment, and the best option is what? The best option is to do culture sensitivity test to find the specific antibiotic which again which can work against the specific pathogenic microorganism, and this is the only best option. Two things is important. One is self treatment is avoided, and more precisely we should do culture sensitivity test to investigate the specific antibiotic against specific infection. This is the best option. Thank you very much.